When it comes to reabsorption in the nephron, there's some simple rules or principles that you need to remember throughout the whole thing. One of them is that water always follows sodium or other solutes when it can. And so we'll be seeing later how some parts of the nephron are not very permeable at all to water and some are very permeable. And if there's a solute concentration gradient, then water can be reabsorbed. And water moves by osmosis when it's being reabsorbed. So we can either squeeze water out of blood vessels under pressure, or we can have it move by osmosis. And for reabsorption, there has to be a solute concentration gradient for water to move by osmosis. And of course, those gradients are established by primary and secondary active transport, which uses a lot of ATP. So make sure that if you are a little shaky on your transmembrane trafficking, that you go back and review that now. And the last thing is, and this will be the basis behind the explanation for diabetes, is that transporters can become saturated. And when I say the explanation for diabetes, I mean the explanation for one of the symptoms of diabetes, which is excessive diuresis or needing to go to the bathroom all the time. First thing you need to do is pull out your notes template of this blank nephron, and you're going to want to make sure that you can label all the parts of this nephron. So filtration, as we already know, occurs in the Bowman's capsule. The filtrate then moves into the proximal tubule, down the descending loop of Henle, up the ascending loop of Henle, along the distal convoluted tubule, and finally down the collecting duct, where it's going to go into the renal pelvis and then move to the ureters, to the bladder, to the urethra, and then, of course, out into the outside world. The next thing we need to add to our diagram is where the cortex and medulla of the kidney are and what the salt gradient is. And just to clarify this, I've included this little inset of the kidney here, and you can see that the nephron, the way it's oriented, the proximal tubule, the Bowman's capsule, and distal tubule are all in the cortex, or the outer part of the kidney here, and that the loop of Henle and the collecting duct extend down into the renal medulla and then drain into this renal pelvis and into the ureter. And it's important to realize that this is a salt gradient, and it gets saltier and saltier as we move down into the center of the medulla. And this is going to become really important when we're talking about reabsorption from these collecting ducts and from the loop of Henle. So now we see we have the adrenal cortex labeled above this dashed line, the adrenal medulla moving down towards the center, and you can see that the osmolarity of this area is actually, it's 300 milliosmolar up in the cortex as well, 300 milliosmolar at the beginning of the medulla, and we end up transitioning, getting saltier and saltier and saltier until we reach a maximum saltiness or osmolarity of 1200 milliosmolar in the center of the kidney. And the shading of this line here indicates how salty it's actually getting. The next thing I've added on this diagram is where the nephron is permeable to water, and these are indicated at the bottom and the top of the drawing. So the proximal tubule and the descending loop of Henle are always very permeable to water. They have aquaporins in their membranes, and if there is a solute gradient, water will cross these membranes. The ascending loop of Henle, on the other hand, is never very permeable to water at all. It's about as impermeable as a biological membrane can get. So water does not have a tendency to leave the ascending loop of Henle. Now, the collecting duct in particular, and at least part of the distal tubule, have their water permeability regulated by hormones. And so we'll come back later and talk about the hormonal regulation about this what a lot of books refer to as the distal nephron. Sometimes they lump the distal convoluted tubule and the collecting duct together and call it the distal nephron. The next thing that I've added on here is the volume of filtrate at each region of the nephron and what the osmolarity of that filtrate is. So when we filter in the Bowman's capsule, we filter, as you already know, 100 liters a day enters that Bowman's capsule, and the osmolarity is about 300 milliosmolar of that filtrate. So basically very similar to the surrounding fluid. By the time we get to the end of the proximal tubule, we are down to 54 liters a day. That means a tremendous amount, well over 100 liters a day, has been reabsorbed along the proximal tubule. Then we move down the descending loop of Henle and go up the ascending loop of Henle, and what you can see is the filtrate now gets saltier and saltier and saltier till it gets to about 1200 milliosmolar, and then it starts to get more dilute and more dilute and more dilute until finally we get to an osmolarity of about 100 milliosmolars, and about 18 liters a day reaches it this far. 100 milliosmolar is more dilute than your body fluids. So your body fluids tend to be about 300 milliosmolar, at least your blood does. Then we move through this hormonally regulated part of the distal tubule, and the osmolarity is still between about 100 and 300 milliosmolar by the time we get to the beginning of the collecting duct. And then as the fluid moves down the collecting duct, we have quite a variable amount of water reabsorbed depending on the state of hormones, which is going to depend on your blood pressure and state of hydration. And we could end up with urine that's anywhere between 50 and 1200 milliosmolar. 50 milliosmolar urine is going to look like water. It's going to be very pale, and you'll be making lots of it. 1200 milliosmolar is really, really concentrated. That's when you're going to have that really dark yellow urine, and you're not going to have very much of it. And if you were to collect all your urine over the day, you would find you would make somewhere between 1 to 2 liters a day that reach the end of the collecting duct. So we're going to work through explaining how all of these changes in volume and osmolarity occur as the filtrate moves along the nephron. The next little bit of anatomy we have to add to this is, of course, the blood supply. So we have blood entering the glomerular capillaries at the afferent arteriole, filtration occurring through the glomerular capillaries. So remember, we said about 20% of the blood enters, moves into this Bowman's capsule and becomes filtrate. The other 80% it moves into the efferent arteriole and then into these peritubular capillaries. And the peritubular capillaries are where the fluid that's reabsorbed from the nephron moves into the peritubular capillaries, and then it ends up going to the renal vein and eventually back into the vena cava and back to the heart again, so it goes back into the circulation. One structure that's a little bit unusual in the arrangement of the peritubular capillaries is this structure called the vasa recta that is draped over the loop of Henle. And the vasa recta is important in preventing the washout of the salt gradient. It's hard to see this the way this drawing has been done, but the blood moves in the opposite direction to the filtrate, and that's called countercurrent flow. And in fact, the whole process of making this concentrated urine and actually setting up the initial salt gradient in the medulla is because the loop of Henle is what's called a countercurrent multiplier. And this has to do with water being able to leave the very permeable descending loop of Henle, but the ascending loop of Henle being impermeable to water and pumping out ions. And this is what sets up that salt gradient in the first place. Maintaining the salt gradient, this passive countercurrent flow is necessary for that. So two different kind of countercurrent processes that are going on to set up and maintain that salt gradient. Just be aware that that happens, but We're not going to ask you to explain that in Bio 277. Okay, so at this point, make sure you have the anatomy of your nephron labeled with the blood supply and what the osmolarities and volumes of the filtrate are. And the first thing we're going to do is look at specifically what's going on in the proximal tubule to result in a large amount of water reabsorption by the time we get to the end, and the filtrate ending up being the same osmolarity. Okay, so focusing in on the proximal tubule. By the time the filtrate reaches the end of the proximal tubule, about 70% of the filtered solutes, so salts mainly, and water are reabsorbed, and 100% of the filtered glucose should be reabsorbed. And this is also true for molecules like amino acids, but we're going to kind of just for simplicity ignore them and focus on the reabsorption of water, sodium, and glucose as an illustration of the principle of how this works. We're going to indicate passive transport processes in green and active transport processes in blue. So the process of reabsorbing solutes, of course, is a property of what kind of proteins are in the membrane. And the first one that we're going to talk about is what we're going to refer to as an always-on sodium pump. So this is basically a sodium-potassium ATPase protein that is constantly using up lots of ATP to pump sodium out of the filtrate. Now, to look at this always-on sodium pump here in a little more detail, keep in mind that these cells of the proximal tubule are not just a single membrane, they're a cell. So if we have the filtrate over here and the blood side over here, this is a proximal tubule cell, and we have to actually move materials into the cell across one membrane, across the cell, and then out of the cell on the basal side. We call this the apical side. And so we can see that we have the sodium-potassium ATPase that we're all familiar with, pumping away to create a very low sodium concentration inside the cell. So now we have a gradient between the inside of the cell and the outside of the cell, and sodium can simply enter by facilitated diffusion, and then it's pumped out of the cell against its gradient to the blood side where it's picked up by the peritubular capillaries. So this always-on sodium pump here that's just drawn as a circle in the membrane is really representing this dual process of sodium moving in by facilitated diffusion, moving across the cell, and being pumped out by active transport. We're calling it an active transport process, though, because it relies on the activity of this sodium-potassium ATPase. The second active transport process we have going on is the reabsorption of glucose. So we have a glucose transporter, and these glucose transporters are also in the membrane. Now, I've drawn sodium on one side and glucose on the other for simplicity, but keep in mind these transporters are all over the membrane here, both of them, and all over the membrane here, both of them. So let's look a little more closely at glucose transport. Well, if you've been reviewing your transmembrane trafficking, you will realize that we have already learned this process. So this glucose transporter that I've drawn here, just as a symbol with an arrow, is really representing absorption of glucose into the proximal tubule cells, movement across the cell, and movement out of the cell again. And again, energy is required for this. We have the sodium-potassium ATPase on the basal side, always pumping sodium out. That creates a sodium gradient where sodium is high out here and low in here. And so that allows glucose here to be moved in against its concentration gradient by secondary active transport because it's using the energy in this sodium gradient. And then once glucose builds up high inside the cell, it can simply leave the cell by facilitated diffusion. So again, we've looked at this when we talked about transmembrane trafficking, and we talked about the jelly bellies and the disruption of that gradient. But we've already worked through this process of secondary active transport of glucose. So absorbed in by secondary active transport, leaves by facilitated diffusion, and relies on primary active transport here with the sodium potassium ATPase to create the sodium gradient. And we're summarizing this whole process here as this transporter symbol. So now we can add water movement on here. Remember, the proximal tubule is always permeable to water. And so now by pumping sodium outwards and pumping glucose outwards, we've now created a solute concentration gradient. And there are other solutes that are moved out as well. And that means that water can now follow these solutes out of the proximal tubule by osmosis. So this is a passive transport, and because it will follow till it reaches equilibrium, the same amount of solute and water leaves the proximal tubule. So by the end of the proximal tubule, we still end up with a osmolarity of about 300 milliosmolar. But we've managed to absorb a great deal of fluid and salts and glucose. This proximal tubule is the only place on the nephron where we have glucose transporters. If any glucose escapes the proximal tubule, for example, if you saturate those glucose transporters, then glucose will move into the descending loop of Henle and be an osmolyte in the filtrate. Because there's no other way for glucose to get out, there's no other glucose transporters anywhere along the nephron, that glucose will end up going into the urine. But the main problem you have, besides losing some glucose, is that now the concentration gradient between the inside of the nephron and the outside of the nephron has now been lost or reduced. So that glucose stays in here, and so the difference between the inside and the outside is not as big as it used to be, so less water is going to be able to be reabsorbed, which is why one of the symptoms of diabetes is polyuria, or excessive urination. Okay, so our filtrate has been filtered. We've reabsorbed quite a bit of water and solutes through the proximal tubule, and now we need to move through the loop of Henle, where we're going to end up with filtrate that's 100 milliosmolar by the time we get to the end, and something like about only 18 liters a day make it this far. We had about 54 liters a day getting to here, and so we've now got quite a bit of fluid that now has to be reabsorbed along this loop of Henle. So let's take a closer look at the loop of Henle. Okay, recall that when we were first labeling our nephron, that we said that the descending loop of Henle is always very permeable to water, so you can see water leaving the loop of Henle. The ascending loop of Henle is not very permeable to water. Also recall that we have a concentration gradient as we move down through the medulla. So we start out at about 300 milliosmolar and end at about 1200 milliosmolar. And so the interstitial fluid of the adrenal medulla gets saltier and saltier as we move down through into the center of the kidney. And so because the filtrate starts out at 300 milliosmolar and it's moving through this saltier and saltier fluid, the membrane's permeable to water, water is going to leave the descending loop of Henle, and in fact the osmolarity of the filtrate is going to reflect the osmolarity of this interstitial fluid around it, because the water will leave till it reaches equilibrium and reaches a maximum concentration of 1200 milliosmolar, because that's the saltiest that the adrenal medulla gets in a human anyway. The ascending loop of Henle is quite impermeable to water, but what it does have that the descending loop of Henle doesn't have is ion pumps. So it uses lots of ATP to pump ions out of the filtrate, so that as the filtrate moves up through the ascending loop of Henle, it gets more and more and more dilute as ions are pumped out, but water is unable to follow. And that's when we end up getting an osmolarity of 100 milliosmolar at the top of the ascending loop of Henle, because we're pumping the ions out, but water is not able to follow. Okay, so now we've shown how the filtrate can get as concentrated as 1200 milliosmolar, and then how we end up getting it to be 100 milliosmolar at the top of the loop of Henle and beginning of the distal tubule. One interesting point is that the loop of Henle is a structure in the kidney that has allowed mammals to produce urine that is much more concentrated than their own blood. So this was a real innovation in being able to conserve water in dry environments. And it turns out that the longer the loop of Henle is, the more concentrated the urine is that an animal can make. Humans, because of the maximum length of our loop of Henle, can only make urine that's 1200 milliosmolar, so that's 4 times more concentrated than our body fluids. But some desert animals have really long loops of Henle and can make urine that's like 20 times more concentrated than their blood. So moving on to the distal part of the nephron, let's look first at the distal convoluted tubule and what's happening along here. So the distal tubule has in its membrane, and keep in mind again, this is a process where ions move across one membrane into these distal tubule cells and then out the other side. So it's still a two-step process, but we're summarizing it as a hormonally regulated sodium pump. So again, this is basically a sodium-potassium ATPase, except its activity is regulated by hormones. It turns out that you guys already know the hormones that are responsible for up and down regulating these hormonally regulated sodium pumps. So aldosterone is a steroid hormone that you guys are already familiar with, and aldosterone upregulates sodium pumping, meaning it causes these sodium pumps to pump faster, and it also causes more of them to be inserted into the plasma membrane. Remember, aldosterone is a steroid hormone that influences protein synthesis. And you also need to keep with you what causes levels of aldosterone in the blood to go up. And remember, aldosterone is produced at the end of the RAAS pathway. So if you forgot about that, make sure you go back and review that, because you still need to know that for this section as well as for the final exam. And you already learned about this in the cardiovascular section. So aldosterone, steroid, has nuclear receptors. The hormone that downregulates sodium pumping is ANP. And so the mechanism isn't quite known, but fewer proteins are in the membrane, so they're lost and their pumping activity is downregulated. And so remember that ANP is secreted in response to atrial stretch, so if you have higher than normal blood volume. So how does this affect fluid reabsorption? Well, the distal nephron, the permeability to it is hormonally regulated as well, and we'll talk about that when we talk about the collecting duct. But ADH will make the membrane more permeable to water, and it makes the collecting duct and part of the distal tubule more permeable to water, and it tends to be secreted at the same time that aldosterone is. So those levels most of the time go up together. And so because the membrane's permeable to water, then water will follow sodium if aldosterone is pumping it out, causing it to be pumped out. At the same time, if ANP is high in the blood, then the sodium will stay in the distal tubule and water will stay with the sodium. And of course, the membrane will be less permeable to water as well. So aldosterone upregulates the removal of sodium from the filtrate. Water follows, so we absorb both water and sodium. So the osmolarity doesn't change very much. And then with A&P downregulates the pumping. And so the sodium stays in the filtrate and the water stays in the filtrate with it. And we get rid of extra fluid volume. Because remember, if we have atrial stretch, then we've got higher than normal blood volume. So you probably drank a bunch of water or other fluids. If the RAAS pathway is activated, your blood pressure is low, and that means that you probably have low fluid volume. You might have low fluid volume, and you need to conserve it. If your plasma gets too salty, if you're too dehydrated, then that actually inhibits aldosterone, because absorbing even more sodium wouldn't be very helpful. Okay, let's look at the last section of the nephron, which is the collecting duct. So the membrane of the collecting duct is characterized by having ADH, or antidiuretic hormone, receptors on it, shown by our purple symbol here. So antidiuretic hormone. And the receptors are along the collecting duct and part of the distal tubule. When ADH binds with its receptor, so if we have some ADH here, so ADH levels go up in the blood, for example, if you're dehydrated, if your plasma osmolarity is too high, or if your blood pressure goes down. The ADH binds with the receptor, and what that does is it causes aquaporins, or water channels, and this is a water channel right here, to be inserted into the plasma membrane. Remember, ADH is not a steroid, it's binding with a membrane-bound receptor. When those water channels are inserted into the membrane and also into the part of the distal tubule as well, the membrane now becomes very permeable to water. And because, if you recall, our salt gradient, starting from the 300 milliosmolar in the cortex to 1200 milliosmolar in the middle of the medulla, so that salt gradient is still there, and this collecting duct is moving down through that salt gradient, then water, of course, is able to leave. And the filtrate could get up to 1200 milliosmolar, but no, it can't get any higher than that in a mammalian kidney. And this is not an all or none thing. The amount of permeability to water depends on how much ADH is being secreted. So if you've got maximal ADH and maximal permeability of this collecting duct, then you will have very concentrated urine of 1200 milliosmolar. You may have some permeability to water if you have a little bit of ADH, so your osmolarity will be intermediate there. Notice that you may have been wondering, well, how can we get an osmolarity of 50 milliosmolar, which is more dilute than it could have started up there? And that's because there's actually some pumping of ions, in particular urea, near the bottom of the collecting duct. And so that pumping out can actually reduce the osmolarity a little bit. And this urea that's pumped out contributes to the saltiness of the medulla. So ADH, when ADH levels are high, this causes water conservation, or reabsorption of water back into the blood again. So figure 25 from Silverthorne summarizes the influence of ADH in a really nice way. So showing on the left side, you can see that ADH levels are really high, so maximal vasopressin allows maximal reabsorption of water back into the blood again. And here you can see that countercurrent flow that I was telling you about, where the blood is moving in a different direction from the filtrate. And of course our urine is very low volume but very concentrated. With no vasopressin at all, then this membrane is virtually impermeable to water, so the water stays in the filtrate and we end up losing it. Once the filtrate has left the collecting duct, it can't be changed anymore. So once it leaves the collecting duct and enters the renal pelvis, we now call it urine, and the composition of it stays the same through the rest of the urinary tract. So without ADH, high volume dilute urine; with ADH, low volume concentrated urine. So I just wanted to end by summarizing the two main ways that hormones can influence water conservation by the kidneys. The first one is influencing how much urine is, or how much filtrate is even formed in the first place. And so filtration is influenced by the state of contraction of the afferent arteriole. And that afferent arteriole is influenced by vasodilators and vasoconstrictors. So remember, we had A and P as a vasodilator, and our vasoconstrictors were ADH, angiotensin II, and epinephrine and norepinephrine. And so when we have a state of low blood pressure, for example, or dehydration, then these are the signals that cause vasoconstriction at the afferent arteriole, and so we have less blood filtered in the first place. When we have to get rid of extra fluid volume, A and P is a vasodilator. The other thing they affect, and I don't think I mentioned it when we talked about filtration, is these hormones also affect the diameter of the filtration slit, so those slits between the podocytes. So I'm going to put filtration slits. So you can imagine, if you want to filter faster, A and P would make them wider, and if you want to filter more slowly, these hormones make them narrower, and that's in fact what happens. The second general thing is reabsorption by the nephron, which is what we just talked about. And so we have, again, 3 different hormones to consider. We have aldosterone and A and P, which influence sodium reabsorption in the distal tubule. And then we have ADH, which affects permeability to water. So 2 general strategies affecting how much blood is filtered in the first place, and then how much fluid and salts are reabsorbed by the nephron itself.